congratulations for winning the Bold and Basic grant.
Thank you.
What is the key question you are aiming to answer? And what would be the best possible outcome?
So, one of the really exciting things in psychiatry in the past couple of years is this rapid acceleration in gene discovery. And so what this means is for the first time we have a reliable set of cues that we can begin to unravel the underlying biology. And one of the ways that we can do that, which what we're focusing on here, is to start with a therapeutics first approach, try and understand the networks and molecular pathways underlying autism, but also the ones that are specifically manipulated by these therapeutics. And so the ideal outcome here is both proof of concept, but also showing that we can alter specific pathways involved in autism pathogenesis, ideally from an atypical towards a more typical state. And so this is really the first step on the path towards developing a targeted therapy.
Nice. Brian, what excites you about this project and how will your lab contribute?
I think I'm most excited about the molecules we're developing for particular targets in the network to which Jeremy referred. Seeing them follow through into the cell biology in Mark's lab and the network biology with Jeremy. And I think that's always our biggest thrill is seeing the molecules penetrate into biology.
Cool. And Mark, what do you look forward to most in this project, and how will your lab complement your collaborators?
Well, we're already actively engaged in collaborations on other projects with Brian, and I've really enjoyed talking with Jeremy as we've been thinking about how to approach this. From my point of view as a cell biologist, the existence of strong genetics now, and increasingly strong genetic evidence for underlying components in psychiatric illness is a super exciting opportunity. And if you think about sort of general physiology, we think of pathways, that's sort of the way that most biochemistry and physiology is organized. If you start looking at the genes that are being identified now with psychiatric disorders, they don't plug in easily to any of the pathways that we presently know. So as a cell biologist, I'm really intrigued by the possibility that these diseases, through this genetic insight, will provide actually information about pathways that we didn't even know existed, that are physiologically important and are deranged in disease. And then between our work and the work with Brian, we hope that we can chemically perturb components in these new pathways with the hope of developing therapeutics that will work in new and improved ways.
Thank you so much. Well, congratulations again, and we look forward to next steps.